Hello friends, welcome to my YouTube channel. Myself Jingle Mystery from Catalyst Classes, Silvassa. In the last video, we had learned about different types of pollination in an elaborative manner. Today we are going to study about the next part of reproduction in flowering plants, that is the process of fertilization. Up till now, we have completed up till all the pre-fertilization events. Today we are going to discuss about the main event, that is fertilization. So the basic need for the process of fertilization will be that the male and female gamete will have to come close to each other, and as a result of which the fertilization process can occur. So pollination, as we have studied till now, is the transfer of male gamete to the female gamete. Right? Now, once the transfer or pollination has been completed, there will be a series of events which will be taking place, and ultimately, after those series of events, what is going to take place is the fertilization process. The first event that we are going to study here is pollen-pistil interaction. So what happens is, on the stigma of a flower, the pollen grain is going to land. There can be many pollen grains falling on the stigma at the same time. Okay? So the event from where the pollen lands on the stigma and pollen tube formation takes place, all those events will be coming under pollen-pistil interaction. Right? Now, once this pollen has landed on the stigma, the next thing that is going to happen is germination of the pollen grain. That means now, the second thing that is going to happen is, first of all, there will be certain stigmatic secretions, and those stigmatic secretions, they are going to activate the pollen grains to form the pollen tube. If you remember very well, pollen grains, they had an outer XII layer, but there was a specific part where it was absent, and through that region, what is going to take place is the formation of the pollen tube. Now, these pollen tube formation, as I said, is only going to take place once the stigmatic chemicals that are released from the stigma are going to activate the pollen grain. Right? Now, so the third thing that is going to happen is formation of pollen tube, which can also be termed as pollen germination. Now, these germination or formation of pollen tube can be monosiphonous or polysiphonous. Now, what happens here is, monosiphonous is the case in which only one pollen grain lands on the stigma, and that single pollen grain is going to form the pollen tube. Whereas in polysiphonous condition, what is going to happen? Many pollens of the same species can land on the stigma of the flower, and out of all those pollen grains, poly is many. Out of all those pollen grains which have landed, only one pollen tube is going to reach up to the egg or the female gamete. Now, when I say polysiphonous condition, that means all the pollen grains which are landing, all of them will germinate to form the pollen tube. But out of all that, only one pollen tube is going to reach up to the egg cell for the process of fertilization. Now, once the pollen tube formation has started, the pollen tube is going to pass through the stigma, right, and style, and further reach up to the female gamete, which is present inside the embryo sac. Now, when we talk about style, there can be two types of styles present. This can be one type of style wherein the style is hollow. Style can be of two types. Two types of styles. One is the hollow style, and The second one is the solid style. Solid style means the one in which the style is completely packed with cells. This is a hollow style, the periphery of which is surrounded by certain cells, right? And the pollen tube which germinates is going to pass through this part, like this. Okay, so this is a hollow style. In case of hollow style, the pollen tube can simply germinate. The pollen tube simply extends through the style, and initially when the pollen tube formation takes place, the nourishment will be derived from the nutrition that is present in the pollen grain. Later on, once the pollen tube has started extending, it is going to derive the nutrition from these nearby cells. Okay. The second style, as I told you, is the solid style. Now, when we talk about solid style, it will be compactly arranged. It will be having cells present in it, which are going to cover the complete surface. So this is the way in which the solid style shows the presence of cell. So now when the pollen tube formation takes place, this is the pollen grain from which the pollen tube has started developing. Now these pollen tube which is forming is basically going to damage, or you can say degenerate, the cells of this type, because the pollen tube which is present here is going to secrete enzymes like pectinases. These pectinases enzymes, they are going to digest the wall of these cells present in the surrounding, and as the pollen tube extends, these cells, they will get digested and the pollen tube get extended cell. So this is the way how pollen tube formation is going to take place in a solid style, and this is the way in which the pollen tube formation takes place in hollow style. As I said, in hollow style, they are going to derive nutrition from the surrounding cells, and in these solid style, they are going to derive nutrition from, again, these surrounding cells. Basically, the cells present here, they are called as they form the conducting tissue. And the process by which they are going to derive nutrition is none other than diffusion. Okay, so diffusion is the process by which in the solid style the pollen tube is going to derive the nutrition. Now, so what is going to happen now when pollen tube formation has taken place? You remember we had discussed earlier, the pollen grains can either be released at 2-cell stage or the 3-cell stage. If it is released at 2-cell stage, which generally takes place in the 60% of the angiosperms, whereas rest of the 40% angiosperms, they release the pollen grain at 3-cell stage. So we are going to discuss now that mostly we are going to consider this case when the pollen grains, they are released at the 2-cell stage. So basically what is going to happen here is, at 2-cell stage when the pollen grain is released, so it is going to have one generative cell and the other vegetative cell, right? Once these pollen has landed on the stigma, the vegetative cell is going to give rise to the pollen tube. So this is the way in which pollen tube formation has started, right? This is the nucleus of the vegetative cell. This is the vegetative cell nucleus, which will be followed by the nucleus of the generative cell. So basically the vegetative cell is going to lead further, where it is going to allow the formation of the pollen tube. What happens next is, since it is released at that 2-cell stage, the generative cell is now going to divide to form the 2 male gametes. So this is the elongating pollen tube, wherein the formation of pollen tube, as I said, is guided by the vegetative cell. We'll draw with this other color. And the generative cell later divides to form the 2 male gametes. So this is the vegetative nuclei and these are the male gametes. Now, when these pollen tube actually forms in a hollow style, there can no error required. There is no error. But what can happen is when it is passing through the conductive tissue, there are chances that because of the nearby cells, these gametes in the nucleus get squeezed off because there are many cells present in the surrounding. So to prevent that, if this is being pressurized by the cells in the surrounding, what can happen? These nucleus can again be shifted back into the structure of the pollen grain. So to prevent that, the pollen tube is going to form a plug, which is called as the callose plug. These callose plug is going to prevent from the contents that are present in the tube to enter back into the upper part. Okay. So this is the vegetative nuclei and this is the two male gametes. The second thing that is going to the next event that is going to take place in the fertilization processes, entry of pollen tube towards the egg, or you can say the female gamete. Right? Now, if I say this is the inside here will be present the embryo sac. These will be the two polar nuclei. These can be the antipodals. This will be the egg apparatus present. Correct? So you can say that this is the micropylar end. This is the chalazal end and these are the integuments. So depending upon from where the pollen tube is entering inside the integument region or towards the egg, there can be three different modes of entry. One is called as porogamy. Porogamy means that if the pollen tube, right, this is the placenta through which it is attached. So here will be the ovary, complete part of the ovary present, right? So if the pollen tube that has entered, if it enters through the micropylar end, the pollen tube, if it enters through the micropylar end carrying the male gametes, then that type of entry will be termed as porogamy. Entry is through micropyle. Micropyle is a type of pore, right? So it is called as porogamy. Second can be chalazogamy. Chalazogamy is the one in which the pollen tube will enter from the chalazal region. And there later on it is going to find the female gamete. So the entry will be through the chalaza. And the last is called mesogamy. Mesogamy is when the entry is through integuments. That means the pollen tube is going to enter from a particular part and reach up to the female gamete through the integument region. Then that will be termed as mesogamy. So these are the three different modes in which the way point of entry from where the pollen tube can come in contact with the female gamete. The last event now, once the pollen tube has reached up to the female gamete, the next process that is now going to take place is actual fertilization. So now when we talk about the last event, that is fertilization process. So let me draw the structure of the embryo sac, right? This is the part of the embryo sac. On one end will be present the synogyns. In between will be present the polar nuclei, and towards the chalazal end there will be the presence of antipodal cells, right? Okay. So now what is going to happen? Let us consider the most common form of entry, that is through the microbiome. So the pollen tube is going to enter from this part. And how they come to know that they have to enter through this part? Because these synergid cells, they are going to have the filiform apparatus, which are going to release chemicals and attract the pollen tube. Here there will be the tube cell, and on here will be the male gametes. Now once the pollen tube enters into it, during the fertilization process, one of the the pollen tube is going to enter into one of the synergid cells. These two are the synergid cells. So when it enters into one of the synergids, this synergid cell will be slowly disintegrating. It will get damaged. The contents of these cells will be lost, right? And the pollen tube is also going to break, resulting in the release of the the two male gametes into the embryo sac. So here what is going to happen? The second stage will be like this. One of the synergid is a bit intact with its nucleus. The other synergid has degenerated. This is the egg cell, the polar nuclei, and these are the antipodal cells. Now the male gametes which are released from the pollen tube, one of the male gamete reaches to the egg cell, whereas the second male gamete is going to reach up to the central cell. This is the egg cell. This is the central cell. So one male gamete goes and fuses with the egg cell, and the other nuclei fuses with the polar nuclei. So basically two fusion is going to take place. One is one male gamete fuses with the egg cell to form zygote. Right? Now these male gamete is haploid, egg cell is also haploid, so the zygote formed will be diploid. Right? The second fertilization process which takes place is the second male gamete plus the polar nuclei. They are going to form the endosperm. Of course, the second male gamete will also be haploid. Polar nuclei. Each nuclei is haploid, but there are two nuclei present, so it will be diploid. And when the fusion of these is going to take place, this tissue which is going to be formed, endosperm, will be triploid. Right? Initially, it is termed as the primary endosperm nuclei, which then later gets converted into primary endosperm cell. And when all the cells are completely formed, it will now be termed as the endosperm. This fusion is called as syngamy, where a male gamete and a female gamete are fusing together. And this, where the second male gamete fuses with the polar nuclei, is termed as triple fusion, because three nuclei are fusing together, so it will be called as triple fusion. So if you see here, 2 times the process of fusion is taking place, or fertilization is taking place within the embryo sac of an angiosperm. So the fertilization process in angiosperms is termed as double fertilization. A very important topic from the board point of view as well as from your entrance exam points of view. So this is about the fertilization process, where there are different events taking place, starting from the pollen-pistil interaction and up to the part where the fusion of the male gametes takes place inside the embryo sac. The next part will be the study of the post-fertilization events. That is now in the next video we will study about what is the role of zygote and what is the role of endosperm. Thank you very much.